Hi students, how are you? I hope you are all good and doing well in your studies. Let's continue the rest of the MCQs of PCR. MCQs number 6. Which of the following is the basic requirement for PCR reaction? Option A. 2 oligonucleotide primers. Option B. DNA segment to the amplified. A heat-stable DNA polymerase, and option D, all of the above. The correct option is D, all of the above. MCQs number 7. Why are Wnt polymerase and PFU more efficient than the Taq polymerase? Because of proofreading activity, because of more efficient polymerase activity, both A and B, and none of the above. The correct option is A, because of proofreading activity. MCQs number 7. Which of the following is the first and the most important step in the polymerase chain reaction? Option number A, annealing. Option B, primer extension. Option C, denaturation, and option D, none of the above. The correct option is C, denaturation. MCQs number 9. What is the process of binding of primer to the denatured strand called? Option number A, annealing. Option B, renaturation. Option C, denaturation, and option D, none of the above. The correct option is annealing. The process of binding of primer is called annealing. Denaturation is a process of heating between 32 degrees centigrade. Option B, heating between 40 to 60 degrees centigrade. Option C, heating between 90 to 89, heating between 90 to 98 degrees centigrade, and option D, none of the above. The correct option is heating between 90 to 98 degrees centigrade. Thanks for watching my video. If you like my video, don't forget to like and subscribe my channel for more lectures. Thank you.